Hi, I'm Madison Strong. And I'm Destiny Woods, and we are biomedical engineering seniors at Grand Canyon University in Phoenix, Arizona. And today we want to present to you our project to create a point-of-care insulin sensor for diabetes. And this project is sponsored by the Helmsley Foundation and Labelle Labs. The problem we are trying to solve with this project revolves around the fact that diabetes mellitus affected 30.3 million Americans in 2015. This is the most accurate data that we can use because after this point, the number of people with this condition is more of an estimate rather than an accurate count. Diabetes is a disease where the body cannot produce or process insulin properly, or the blood glucose levels are not maintained as other people's bodies would maintain them. This project is focusing on type 1 diabetes, which is the inability of the body to produce insulin. And this number, the 30.3 million people affected with diabetes, does not account for other countries. So obviously this is a widespread problem, and it's only expected to continue growing as years go on. Glycemic profiles, which is what a person with diabetes would be monitoring when they're doing blood sample testing, is currently only monitored by one biomarker, but would actually be better monitored by multiple biomarkers such as insulin and glucagon. Current methods, as I said before, need advancement, and currently only glucose is used, but it's also used for insulin dosing. So we need a more direct approach to insulin dosing that uses insulin levels rather than glucose levels, so that a person with diabetes can make more informed decisions about their health. Finally, the only current options for insulin testing require special laboratory facilities, trained staff, and many hours to complete, which is not adequate for a test that needs to be done in under a minute for someone to be able to make informed decisions about their health. Our solution to this problem is to create a biosensor that measures insulin concentrations rather than glucose concentrations. Essentially, the sensor would use electrochemical properties of a chemical layer applied to an electrode surface and would measure the electrical properties of a chemical reaction which occurs after the placement of a sample onto the surface that includes insulin. This is similar to how glucose testing is done with the current glucose test strips. However, we want to focus again on insulin so that people with type 1 diabetes have more accurate data on how to dose their insulin. Again, this project is funded by the Helmsley Foundation, and it is a total of a 3-year grant, with the first year being to create a reproducible sensor that uses purified insulin. And purified insulin is not from a blood sample, it is just insulin that comes from a lab. The second year is to use insulin from blood samples, so this would require an additional coating on the sensor surface, and then we would transition into an animal study so that we're using live blood samples rather than those from a laboratory. And finally, the third year is to combine the data and processes that we've learned and used to measure insulin samples with those used to measure glucose samples so that a dual biosensor can be created.
The main purpose of this year is to develop a reproducible protocol. The most significant way of testing the consistency of the protocol is by developing a standard curve. This curve is produced by comparing the tested concentration range to the electrical properties from those concentrations. Once the standard curve has been replicated multiple times, we will then be able to determine the unknown concentration by comparing it to the electrical properties found on the curve. The desired curve has an R-squared value of 0.95 or higher, which demonstrates that the protocol has produced sensors with little variability. This is optimal to ensure that we can trust the process that has been developed, and we can trust that it displays both accurate and precise results. Accuracy and precision are both necessary in providing our customers with data that they can rely on regarding their glycemic profile. The next major desired outcome is an optimized protocol. A baseline process has been developed, but it leaves room for a significant amount of errors. The main source of error thus far has been operational, where each step varies depending on who performs it. This difference produces inconsistencies in the sensor development, which then alters the data. Within this optimized protocol contains exact measurements of any chemicals, including volumes and concentrations. A great deal of research is being performed to determine what these values are in order to produce optimal results. The amount of time that each chemical layer rests on the sensors is also important, as it determines how much of each molecule will be bindable. We hope that we are able to develop a protocol that greatly decreases the potential for error, which improves our chances of making sensors that are consistent in their results.
The design process thus far has consisted of a series of immobilization experiments. These experiments take roughly 10 to 12 hours over the course of 2 days, with a 48-hour gap between these 2 days. So it takes quite a long time to conduct these experiments, so our team only conducts about 1 experiment per week. Each experiment varies in that we are able to observe one variable and see if it has a significant effect on the end results, such as the accuracy and the precision of the sensors and the protocol. The first part of the experiment involves adding a series of chemical layers to an electrode base. This series can be found in the figure to your left and is widely used in research and can be used to determine the concentrations of different biomarkers. However, we are adapting this protocol so that we can only measure the concentration of insulin. After these chemical layers have been added, we are then able to test a series of concentrations of insulin, and the range that we are testing is what is found clinically. So this is what's found naturally in a human body, and we are also testing additional concentrations so that we can compare how our results are in terms of precision and accuracy and compare those to the current insulin monitoring techniques such as ELISA. And after we have tested these concentrations, we are then able to analyze the data. This produces the standard curve that was talked about previously, and it is able to tell us how the accuracy and precision of the protocol and the sensors have been, and if we need to improve the protocol further. And so we do a series of tests measuring one variable at a time. So once the first variable is optimized, then we can move on to the next variable and so on. Although we have worked so diligently in the first part of this project, there are still 2.5 years left in the project lifespan. Currently, there are 3 teams working under the guidance of Labelle Labs. The first team, which is our team, has worked on the optimization of protocol while also creating a reproducible chemical layer that can be applied to sensors so that we can run the proper electrochemical impedance test. The second team is working to verify the protocol that we create while also researching how to coat the sensors for the application of blood samples, which will be the focus of year 2. And finally, the third team is also verifying the protocol we create while testing if there's an overlap between glucose and insulin concentration curves so that we can find distinguished differences between them so that we can make a dual sensor for each biomarker. Over the next coming months, we hope to transfer our collected data to the other teams while also preparing them for using our protocol and transitioning over to year 2, which officially begins in July of 2020, which will then focus less on the optimization of the protocol and more on the application of a blood sample, including insulin, rather than the purified insulin samples. Our team will also be focusing on preparing for a publication so that we can publish our findings from year 1. For your reference, here are our works cited. We are unable to disclose any further information about our project because we are still working under the Helmsley Foundation. We would like to thank the Helmsley Foundation and the Labelle Labs for allowing us to conduct this research, and we also thank you for your time and hope you and your families are staying safe.